Alverno is sponsored by the Franciscan Alliance and Presence Health Systems. The central laboratory is located in northwest Indiana, delivering services to its hospitals in Indiana and Illinois. It serves 8 million people in need of diagnostic testing. Join us on a tour of the main lab. We begin our tour at the loading dock. We will follow 4 samples through testing. First we drop off at General Lab. Now we drop off at Micro. Next we drop off at Anatomic Pathology. And finally we drop off in our molecular laboratory. Now let's go back to the General Lab to see the testing process. The General Laboratory Department performs over 300 different diagnostic tests and operates 24 hours a day, 7 days a week, 365 days a year. Over 40 state-of-the-art analyzers perform and analyze sensitive diagnostic tests. It is the medical technologist's job to interpret the results and make sure that each result is handled with high standards before releasing the results to the ordering physicians. Alverno serves several thousands of patients each and every day. Patient care is our top priority. The specimen enters the automated line, the Beckman Power Express. It is a state-of-the-art automation line that can handle 1,200 tubes an hour. It receives the sample in the laboratory information system, centrifuges the sample, aliquots the sample, and routes the sample to the appropriate analyzer for testing. It has the option to archive the completed specimen in the automated stockyard, and when a physician wants to add on a test to that sample, it can automatically retrieve the sample for further testing. Once a sample is placed on the automated line, it does not have to be touched again. As a sample moves along the line, it already knows its destination. It may proceed to the automated hematology area if a complete blood count is ordered. The science of hematology may help the doctor find an infection or diagnose a patient with anemia or leukemia. As we visit the hematology area, the specimens arrive at the rack builder and are placed in the next open location in the rack. When the rack is full, or when 60 seconds have elapsed, it will enter one of the 3 DXHs. If some of the result parameters are not met, the specimen will travel to the slide maker stainer, where a slide is prepared from the patient's sample, and a technologist will prepare a manual differential, which generally is the counting of types of white blood cells, looking at red blood cell morphology, and estimating platelet counts and sizes. Typically, about 10% of CBCs require a manual differential. If chemistry testing is needed, then the sample will go on to one of our DXI 800s or one of the AU 5822s. These analyzers are fast and robust and execute a large volume of tests in a short amount of time. Moving probes dip into the patient sample and mix in the required chemical reagents in accord with the test ordered. Once thoroughly mixed, the specimen appears before a specialized lamp to record absorbance, readings, and calculate a result. 2 Siemens Centaurs are connected to our automated line, allowing the flexibility to perform infectious disease and tumor market tests in a real-time fashion instead of batch testing, thus improving turnaround time. Regardless of where the testing is done on the line, the technologists utilize Remisol to interpret and verify results. Remisol is the middleware between the analyzers and our laboratory information system. From a single workstation, techs release valid results to the LIS, which proceed to the patient's chart and enter the physician's electronic medical record system. Our LCMS, liquid chromatography mass spectrometry system, is an analytical chemistry technique that combines the physical separation capabilities of liquid chromatography with the mass analysis capabilities of mass spectrometry. LCMS is a powerful technique that has very high sensitivity. transplant drug testing occurs on this platform. Other methods of testing in the general lab department provides results as varied as counting and identifying the cells in blood and body fluids to determine what your child might be allergic to the risk that an unborn child may have certain birth defects. Physicians diagnose diseases as varied as diabetes and leukemia, infertility and vitamin deficiencies, gluten sensitivity and cancers via diagnostic testing. The general laboratory is truly a remarkable place, and it requires a team effort of dedicated individuals to help provide excellent care for our patients. Microbiology at PCL Alverno provides comprehensive state-of-the-art infectious disease-related analysis of many different types of cultures, including bacterial, fungal, and tuberculosis. Over 2,000 cultures are evaluated each day by highly trained professional medical technologists. When a specimen arrives in microbiology to be processed for culture, it will be placed on the walk-away specimen processor, otherwise known as the WASP. The specimen is picked up by a robotic arm to scan the barcode, which retrieves vital information about the test from the lab computer base. The next stop in the instrument is where the specimen is mixed and the cap is removed. Another robotic arm dips a sterilized wire loop into the specimen and transfers a specific amount to different types of media plates. These are streaked in a specific pattern to spread out any bacteria present. The plates are moved to a station where a barcode label is applied to the side of the plate. Before leaving the machine, the label is scanned one last time to assure the accuracy of the completion of the sample setup. From the WASP, the plates travel on a conveyor belt to the WASP lab incubator, where once again the plates are barcode scanned to assure specimen accuracy. Before they are placed in the incubator, a high-resolution camera takes a picture of the surface of the plate as a reference to compare to future pictures. After posing for a photo, the plates are transferred into the incubator, which is maintained at the temperature, humidity, and carbon dioxide level for optimum bacterial growth. The incubator is programmed to remove the plates at specified times to detect changes in the surface of the media that would indicate the start of growth of bacterial colonies. Cultures that do not produce growth after a specified time of incubation are automatically reported as no growth by the WASP lab system without technologist intervention. Once growth is detected, the technologist will be alerted via computer that a positive culture is ready to be worked on. The technologist will view the images at their workstation and decide which colonies need to be worked up for identification and antibiotic resistance testing. Using the mouse on the computer, the specific colonies will be chosen and highlighted for identification by a piece of equipment known as the Malditoff, and for antibiotic-resistant testing by microscan prompts. The WASP lab incubator will retrieve the plates that are needed for Malditoff and prompts and stack them in a canister for a technologist to pick up and transfer to the WASP. Accessing information from the WASP lab computer, the WASP will highlight the colonies from the appropriate plate and transfer it to the Malditoff target circle and inoculate the prompt bottle for microscan antibiotic resistance testing. The target is loaded into the instrument and run. Once inside, a laser is used to vaporize the bacteria, and the different protein molecules travel up a vacuum chamber to a detector at the top. The amount of each type of protein is determined and compared to a database of over 2,000 bacterial species to find the best fit pattern and identify the organism, giving it a name. The prompt bottle is taken to a workstation to transfer it into a 96-well panel containing 26 different antibiotics. This panel is placed in the microscan instrument that incubates it for 18 hours. Each well is checked for growth of the bacteria to determine which antibiotics kill the bacteria and which ones were not effective. The results of the culture go through a final evaluation by a technologist before they are released electronically to the physician. The availability of this breakthrough technology prompted us to centralize blood cultures from the hospital sites to the central laboratory. Samples are transported inside custom incubated transports and placed on one of 12 Bactec FX units. On any given day, there are an average of 4,000 blood culture samples in process. The MALDI Sepsi Typer kit is also used in conjunction with the MALDI-TOF for direct pathogen identification from positive blood cultures. At the time of positivity, an aliquot of a blood culture is processed using the MALDI Sepsi Typer kit. Harvested microorganisms from this process are then spotted onto the MALDI-TOF target for identification. The MALDI Sepsi Typer has proven to reduce turnaround times compared to traditional workflows by 36 to 48 hours for pathogen identification from positive blood cultures. The function of microbiology is threefold: to identify if there is an organism causing infection or not, identify and name the organism, and provide information of which antibiotics will be effective. The overall result will be the potential for physicians to use antibiotics with a more narrow spectrum when treating infections. This helps reduce the probability of development of highly resistant strains of bacteria, possible shorter lengths of stay in hospitals, and reduced healthcare costs. Every tissue specimen that is surgically removed, from small biopsies to whole organs, will find its way to a histology department. These specimens are examined either by a pathologist or a pathology assistant in a procedure called grossing. The specimen is examined for any abnormalities, and either samples from the specimen or the entire specimen are submitted for processing. Processing tissue samples consists of putting the samples through several changes of different types of reagents in order to ready the tissue for embedding, sectioning, and slide preparation. Embedding the tissue refers to orientating the biopsy into a paraffin wax in such a way to allow the structures of the tissue sample to be sectioned by the histotechnician from a paraffin block. The embedded tissue is then placed onto a microtome, and the histotech sections each block by obtaining paper-thin paraffin ribbons that are placed onto a warm water bath. From the water bath, the thin sections are picked up with glass slides, and the slides are then placed into an oven to prepare the slides for staining. Histology uses automated stainers to perform the routine H&E stain required for every slide cut in histology. The H&E stain will stain the structures of the tissue different colors, allowing the pathologist to diagnose the patient biopsy. After the slides are stained, the slides are placed together to complete the patient's case. The slides are delivered to the transcription area where the gross description has been typed into a report. That report and the patient's requisition and history are placed with the patient's slides and given to a pathologist for microscopic review and diagnostic interpretation. The Molecular Biology Department is a multidisciplinary laboratory that is dedicated to providing patients and physicians with comprehensive and accurate molecular testing. Our laboratory uses progressive technologies and procedures such as real-time PCR, fluorescent in situ hybridization, and flow cytometry to evaluate inherited disorders, malignancies, and infectious diseases. Real-time PCR technology is used to quickly and precisely detect pathogens such as bacteria or viruses in a patient's specimen. This is done by identifying a unique piece of the microorganism's RNA or DNA and making enough copies of that piece so that it can be easily detected. Test panels can be performed to identify many pathogens that can cause respiratory infections, wound infections, gastric infections, cervical infections, or blood and spinal fluid infections with comprehensive results in a few hours. A patient's sample could also be tested to ensure they do not harbor an opportunistic microorganism such as MRSA that could cause severe complications in an immunocompromised patient or a patient about to undergo surgery. The molecular department also performs viral load testing for HIV, cytomegalovirus, and hepatitis viruses to assist the physician with treatment of their patient using antiviral drugs or immunosuppressant drugs. Our fluorescence in situ hybridization, FISH lab, offers comprehensive panels for diagnosis, prognosis, and disease monitoring of many types of cancers, such as leukemias, lymphomas, bladder cancer, lung cancer, breast cancer, and gastric cancer. Here cells from blood, bone marrow, or tissue are placed on a slide and treated with DNA probes specific for genetic targets. These probes will emit a fluorescence color that can be analyzed by state-of-the-art imaging systems and examined by highly trained and certified technologists. FISH analysis can identify deletions, amplifications, and rearrangements of genetic material in a tumor specimen. The identification of these mutations provides information to the physician to diagnose or treat a patient's tumor. Our flow cytometry laboratory complements FISH testing for the evaluation of leukemias and lymphomas. Here cells are placed in a fluid suspension and incubated with monoclonal antibodies. After washing the cells to remove unbound antibodies, the cell suspension is run on the flow cytometer for analysis. Our newly acquired 10-color Navios flow cytometer is capable of measuring the types and quantities of bound antibodies on up to 20,000 cells per second. After the sample is ran, a sophisticated data analysis software is used to analyze the information and generate plots, statistics, and visual representations of the types of cells found in the patient sample. This information is then reported to the pathologist for review and correlation with other laboratory and clinical data. The final report will assist the physician with the treatment of the patient. Cutting-edge technology and innovation contribute to the description of Alverno as a highly reliable, scientific organization building a culture of service. Alverno is made up of people who work for a healthy tomorrow. Alverno personnel demonstrate courage, awareness, and perseverance as a platform for continued success in the service of the sick.